Good morning, good morning, viewers. Welcome to MouShauk. Today, I'll be discussing the large intestine and the liver. Okay, the liver, the functions and composition. Okay, and assimilation of food also. So before I start, I will request you to please subscribe my channel if you have not subscribed yet, or if you are a new viewer to my channel. And if you are liking the video, press the like button, share with your friends so that they can also be benefited from this video. And do meditate before you start your day. Okay, so we will also start with a short meditation, and then we will start our discussion. Okay, so for meditation, let us concentrate on the point of light. Think that light is falling on you. Your whole body is being enlightened. You're feeling very light without any problems, without any heaviness. You're feeling as light as very. You can fly in the sky, just like your body is. You're feeling all problems, all tensions. They're so light and tension free that you can enjoy every moment of your life. And you can, whatever you are given, you can do it without any dissatisfaction, and they're enjoying your work, they're getting interest in your work, and at the end of the day, you're becoming successful. So you're thanking God for being with you, with your side, and taking care of every step for you. Thank you, God. And I thank for the good things. Okay, in the last video, I told about the bile duct and the pancreatic duct. Okay? So this was the picture which we last ended, that this was the gallbladder, and the hepatic duct is coming from the liver. The gallbladder, the duct which is coming from the gallbladder is the cystic duct. Both of them are joined together to form the common bile duct and entering into the duodenum. From the same aperture, through the same aperture, the pancreatic duct also enters into the duodenum from the pancreas. Okay, pancreas is the yellow leaf-like structure present in the left-hand side of the body of the abdomen region. Okay, so it is there, and the duodenum is the part of the small intestine below it. Okay, so now let us move on to the large intestine. So it was the small intestine part which we discussed in my last video. Now we are coming to the large intestine. The large intestine is 1.5 meters long. Okay, and it has main three parts. One is cecum, second is colon, and third is rectum, which is ending in an anus. Okay, ending in an aperture outside the body, which is known as anus. Okay, now cecum. Cecum, what is cecum? It is a small blind pouch, okay, which is situated at the junction of the small and the large intestine. Between the small and the large intestine, there is a small blind pouch, okay, which is known as cecum. Okay, and it is from its blind end projects a narrow worm-like structure. Okay, worm-shaped tube-like structure, which is known as vermiform appendix. Okay, it's a vestigial organ for humans, but in other animals, it is functional. Okay, but for humans, it is a vestigial organ that does not require, we do not have any function of this. And if it is inflamed, it causes appendicitis, and it's a great pain which one experiences one if one has this appendicitis. Okay? So this is the part of the cecum. Now we move on to colon. Colon is the much broader part of the intestine after the ileum. Ileum is very narrow part, which after that comes the colon, and between the colon and the ileum is the cecum. Okay, and when the colon starts, it first is the ascending colon. It first starts with an ascending colon, then transverse colon, and then descending colon. Okay. Now the length of this part, this colon part, is a little more than a meter long. Okay. So it will be ascending, then transverse, then descending colon. Then we come to rectum. After the colon ends, that is the descending colon, after the descending colon ends, the last part of the large intestine is the rectum, okay, which is about 15 centimeter long. And it opens outside the body as anus. Okay. Now, what is anus? Anus is the opening, we know it, that of the rectum outside the body, but it has some sphincters which keep it closed, and it opens only when we require to pass the bowels. So it opens to pass the bowels. That time only it opens, otherwise it remains closed. This is done by what? It is done by the sphincter. Okay. Some muscular rings, sphincters are nothing but muscular rings which are present in certain in the certain spaces. Okay. Like we found in the esophageal sphincter, then pyloric sphincter we found it. Okay? So these are all muscular rings which are present to prevent the backflow of the substances, or you can say to regulate the flow of any substances in one direction. Okay. Now, functions of this large intestine is it has no enzymes. Main thing, it has no enzymes, and here digestion is not done. Or rather, you can say very little digestion is here done. Okay, mainly here absorption takes place. Mainly absorption of water takes place, and very little digestion food. Very little digested food is digested here. Okay, consists mainly of undigested material here. Okay, after much water absorption, the contents which remain, that is almost semi-solid, which is known as feces, we call it. Okay, that pass into the rectum and are expelled at intervals to the anus, which is known as defecation. So this process of eliminating or rather expelling the feces out of the body at regular intervals through the anus from the rectum is known as defecation. Okay. Now, composition of feces. Feces, what is the composition of feces? Feces has 75% of water, 25% of solid matter, which is composed of 30% dead bacteria, 10 to 20% fat, 2 to 3% protein, and 30% of lipid. So mainly two things are there. One is water, another is solid matter. Okay, 75% is water and 25% is solid matter. Now, out of this 25% of solid matter, 30% is dead bacteria, 10 to 20% is fat, 2 to 3% is protein, and 30% is lipid. Clear? Now, we know that this feces have a bad odor. Okay, why is it so? Due to the bacterial action in it. We know they have 30% of dead bacteria. So dead bacteria and also some bacteria acts on it to give that bad odor. Now, the composition of this species varies from person to person. Why? Because of the kind of food they consume. Okay, if they take more of protein, their composition will be something different. And if they take more of fat, then the composition of the feces will be something different. So depends on the kind of food they consume, the composition of the feces varies. Now, This food which is being absorbed by the intestine is now assimilated. Okay, now what is assimilation? Assimilation is conversion of the absorbed digested food into the bodily material. Okay, so that is protoplasm. We have to convert the food that is absorbed, the digested food that is absorbed to the protoplasm, which is the body material. Okay, so for that conversion, a process is required, and that is what assimilation, that is called assimilation of food. Now, how is it done? First, the digested food is absorbed by the gut or the alimentary canal. Okay, so the alimentary canal, it absorbs the digested food. Now that is transported by two different ways. Okay, transported in two different ways. One is hepatic portal system, another is lymphatic system. Okay, hepatic portal system directly takes the digested food, the absorbed food to the blood. Okay. And lymphatic system, first they are taken by the lymphatic vessels and then to the blood. Okay. Now, which food are taken by the, the hepatic portal system and which are by the lymphatic system? Let us see. First, the simple sugars, amino acids, vitamins, minerals are carried by the liver. That is by hepatic portal vein to the blood circulation. So what are those foods which are taken by the by this hepatic portal system? One is simple sugar, amino acids, vitamins and minerals. Okay. Now lymphatic system, they take what? The fatty acids and glycerols, which have been absorbed by the gut. Now let us see hepatic portal system, how they take this food. Okay. So these are done by two different ways. Not two different ways. It is two different kinds of food are taken by the hepatic portal vein. Mainly, vitamins and minerals are directly absorbed into the blood. Okay, by the hepatic portal vein. But simple sugars and amino acids, they are more simplified and then they are being absorbed. Now, excess glucose. What happens? Excess glucose by glycogenesis. Glycogenesis is a process by which excess glucose is converted to insoluble glycogen and stored in the liver. Okay. Temporarily stored. Okay. And when required, when the body requires glucose, then by glycogenolysis, this is another process by which the liver gives back the glucose into the blood. Okay. So, reconverts the glucose from the liver to the blood circulation. Okay. Now, these are the two processes which happens in the liver. One is glycogenesis, another is glycogenolysis. Glycogenesis is excess of glucose to insoluble glucose and storing. And glycogenolysis is storing from the stored glucose to soluble glucose into the blood. Okay. Now, glucose, what does glucose give? They give the body tissues a source of energy in cellular respiration and synthesizes certain compounds. So, this is taken by the hepatic portal vein to the blood circulation. Okay. And it reaches the cells and it gives energy to the cells in cellular respiration, and also it also synthesizes certain compounds. So bodily material it has been transferred to. This is glucose. Now to amino acids. Amino acids circulate in the body and they serve serve as the building blocks of protein. Okay, amino acids are the end products of protein. So they give the building blocks of protein, or you can say the brick of protein. Okay, after that, layers of amino acids, we can build the protein, whole protein. Okay, so this is the unit of protein, we can say. Now, by deamination, excess are broken down into liver, broken down in the liver, and the nitrogen which contains the amino group is removed and converted into urea for excretion. The remaining part forms the glucose, which can be utilized. See, from amino acids, again, glucose can be converted. So, excess of amino acids, from the excess, the amino acids, if say a person takes more of protein, so it contains excess of amino acids. Okay, that excess of amino acids, on that there is a process which is applied on this excess of amino acids to form, to break down this amino acid into nitrogen containing amino group and the remaining part to glucose. So the nitrogen containing amino group is removed and converted into urea, which is excreted. Okay, and the remaining part which forms the glucose is utilized by the body. But this process of breaking down of amino acids into nitrogen containing amino group and the rest of into glucose is known as deamination. Deamination is the process. Okay. Now, this one to the lymphatic system. Lymphatic system takes what? The fatty acids and glycerols which are absorbed by the alimentary canal. They are transported mainly through the internal villi and, intestinal villi and lymphatic system. Through the intestinal villi and the lymphatic system, that is the lacteal which is present in the villi, that carries this fatty acids and glycerol. Okay, they are transported by that into thoracic duct of the lymphatic system. From there to the large veins to the heart, and some fats are used in the synthesis of certain compounds in the body cells. Okay, some fats are used for synthesizing certain compounds in the body cells, and excess quantity, excess of fat which is present, that is fatty acids and glycerol which are present in excess amount, that is deposited chiefly in the bottom of the skin, chiefly under the skin as subcutaneous fat or around certain visceral organs in small quantities. In small quantities, they are stored, they are covering the visceral organs, around the vertebral organs, they can be stored or they can be deposited just under the skin as subcutaneous fat. Okay. Then this is about the liver, the large intestine absorption and assimilation of food. Now we come to the liver. What is liver? Liver is the largest gland of the body. Okay, its weighs about 1.5 kg on an average. Average weight is 1.5 kg. The color of this organ is reddish brown. It's located in the upper right side of the abdomen. First, just below the diaphragm. Just below the diaphragm, in the upper right-hand side of the abdomen is the liver. Okay. Now, what are the functions? We know main function of the liver is production of bile. We have studied bile earlier. Now, the rest of the functions we will study. Okay, besides the production of bile, it also controls blood sugar levels, controls the amino acid levels. It synthesizes fetal RBC. Okay, it stores iron, copper, several vitamins, etc. It detoxifies drugs and alcohols. Okay, it produces fibrinogen and prothrombin. It produces heparin. It produces heat by cellular metabolism. It regulates blood volume by acting as a temporary store of the excess water. Then excretion, which also helps in excretion of toxic and metallic poisons, even it destructs the dead RBC. So, first is it controls the blood sugar level. By which process? By glycogenesis and glycogenolysis. Like glycogenesis, it will convert the excess sugar in the blood to the insoluble form of glucose and store it in the and when it is required, when the body requires glucose, it will revert back the glucose, the stored glucose into soluble glucose by glycogenolysis. Okay. Now, controlling of amino acid level by deamination. The excess of amino acid which is present in the blood, that will be broken down by deamination. Nitrogen amino group will be converted to urea and excreted from the body, and the the rest part of the amino acid will be converted to glucose and will be used by the body. Then it synthesizes fetal RBC in a fetus when a child is in the womb of the mother. At that time, the RBC of the baby of the child will be constructed in the liver, built in the liver. Now it stores certain minerals, that is iron, copper, and several vitamins like A and D. Okay? It detoxifies drugs and alcohol with others. Okay? So others also detoxification, other detoxifications also take place, but drugs and alcohol is also detoxified in the liver. Now, production of fibrinogen and prothrombin, which are required for blood clotting. Okay, fibrinogen and prothrombin are formed in the liver. The production of heparin, which is an anticoagulant, which coagulates blood when you have a cut, no, the blood starts flowing out. So at that time, heparin, an anticoagulant is required. A coagulant is required. Okay, that is different, but that is okay. But when the blood is flowing inside our body, in the vessels, the blood will not clot inside our blood. Inside our body. So that is because it is due to the heparin, which is an anticoagulant present in our blood, which is produced by the liver, which helps to not to clot the blood inside our body, where the blood is flowing inside the vessels. Okay. Then production of heat by cellular metabolism. When cell metabolism takes place, the heat is produced. So that heat is produced by the liver. The production of blood volume by acting as a temporary store of excess water. So when we take water, okay, we drink water, take water through different sources like through food which we eat, through the fruits, the vegetables. Okay, so water is going inside our body. So excess water will be stored in the liver to maintain a certain volume of blood in our body. When the blood volume is becoming less, that time the excess water will be released. The stored water will be released inside the blood so that the blood volume maintains a required volume inside it. So the blood will maintain a required volume inside our body. Okay, excretion of toxic and metallic poisons. Okay, some toxic and metallic poisons are present in our body, which needs to be excreted. That is done by liver. Then dead RBC, the RBC which are becoming dead, which are present in the blood, that is destructed by what? That is destructed by our liver. Okay. So this is for today. So next part will be some tests for starch, for protein. Okay. So we will discuss, these are the practical work which has been discussed in your book. So we will discuss in that chapter, from that chapter. And next, after that, we will go on to the question answer section of this chapter. Okay. So requires some time. Please do bear with it. Okay. And do subscribe if you have not subscribed yet. And if you are a new viewer to my channel, and please do meditate before you start your day. So that will help you in your concentration power, increasing your concentration power, increasing the interest in your studies and getting the marks over. Okay. So thank you for joining and have a good day.